Well, we said when we started this transmission genetics series that there were two sets of lectures that were really important. The first one we've gone through, the monohybrid crosses. Very simple. I know you've seen it before, but hopefully you've been refreshed on it. And most importantly, hopefully you have Mendel's Big Six. You're going to need them for this as well, but you have certainly learned all of these crosses, the six crosses, and their immediate outcomes, both genotypically for Mendel, phenotypically as well. But genotypically is very, very important. So I have mine in front of me. I hope you have yours in front of you. So let's take it and let's just take the basic information, take the Mendel's Big Six, and we're going to just add a little bit of variant to it, okay? Remember what Mendel had found to this point was that the genes in these peas occurred in pairs. In other words, they were diploids. And he described what we were calling our first mode of inheritance, which is dominant recessive. Basically, when you get a heterozygote, the cell has one allele, basically takes over and decides what's going to happen, right? Again, being anthropomorphic, but essentially saying that at the molecular level, if you've got two pieces of genetic information, one of them will dominate over the other when you have a heterozygote, okay? And then, so those were ideas. But then we said, well, there's this, we're going to call this important next thing a principle. Again, there are not many principles in biology. So when you see something called a principle, it's very important. So the principle of segregation, that's dealing with monohybrid crosses, and basically says during formation of gametes, during meiosis, these alleles that you have on these homologs will just separate randomly. You can't predict which gamete they're going to go into. Okay, so they're going to do that. All right, we're going to take that principle that's happening here and we're going to make an advance on it, okay? Because what Mendel did was obviously he looked at each one of these traits that we found here out of his 34 that he total that he had, and he would make crosses with smooth versus wrinkled and take them to the F2 and this. But, you know, he didn't have time to do every one of them all the time, so being very smart, he would do multiple ones at a time. He would take maybe the seed shape smooth and wrinkled and maybe pod color green and yellow, and he would put those together into one cross. Well, we would call that a dihybrid cross. And this is a cross that involves two traits or more. And it can be 3 traits, 4 traits, 5 traits. It would be then a multi-hybrid, right, at that point. So you'd have 3 traits would be a trihybrid, and then you have multi-hybrids. But we're going to concentrate on 2. So we're going to use a dihybrid. Notice there's a little star right there by it, okay? That little star is there because we're going to find out that you don't necessarily have to have 2 traits sometimes to have a dihybrid cross. What you have to have are 2 loci. Okay? For now, don't worry about that, but I just want to put that there so when we do something later, we don't have an error in it, okay? All right, now we've got our classic parentals. This time though, we've got 2 traits, remember? So we've got smooth and green because we picked those, and they go up against wrinkled and yellow, okay? Now, once again, Mendel's Big Six. Nothing's going to happen that's not explained here. All right, we're going to make these crosses. We're just going to make multiple ones at the same time. So in a parental is just going to be 2 of these together instead of 1 of those together. And when we do other crosses, the F1, it'll be 2 of these together instead of 1 of those together. All right. So if we take a look at that, remember that Mendel was smart enough to use true breeding, and that holds here. He was able to pick homozygous genotypes for both his smooth greens and for his wrinkled yellow peas. He was very careful to do that. We'll just designate them. You could make them big A, big B if you want. Doesn't matter to me as long as you keep them consistent. We're just going with some other letters here, change it up for a bit. Then we make our gametes, right? And this is a homozygote, so we can only make one kind of gamete. The genetic information that it'll pass on carries big S and big G. And if we go over here, this one's only going to pass on a little s and little g, wrinkled and yellow. Well, when we cross those, of course, make the first biallel generation, everybody gets a big S and a big G from this parent, and everybody gets a little s and a little g from this parent. Very straightforward. So you're going to have a heterozygote. It's called a dihybrid heterozygote. So dihybrid heterozygote is dihybrid because there are 2 loci. If it were 3 loci, it'd be a trihybrid heterozygote. But this is called a complete heterozygote or a dihybrid heterozygote. And we've got a heterozygote at the first locus, the S locus, and we are heterozygous at the second locus, which is the G locus, all right? Because of dominant recessiveness, we'll just say that smooth is going to be dominant over wrinkled and green is dominant over yellow, which they were for Mendel. So these would all be smooth green, right? Then being Mendel, we would make our F2 cross, right? We would produce the F2 progeny by making this F1 cross. So we're going to self these individuals if we could, or if we're doing animals or something, we would just find litter mates with the same genotypes or something and cross those. All right. Well, now we've got slightly more complication because now to produce this F2 generation, we've got more options, right? We can produce a gamete that, because these separate, we can produce a gamete that carries an S and a G, big G. That's one kind of gamete. We could get a gamete that has a big S but picks up a little G. We could have one that has a little s, in other words, a wrinkled piece of mutation or genetic code while carrying the green allele for the other locus, okay, big G. Or we could get some that are going to produce, wrinkled and yellow because we're carrying that up here. Now at the same time, this one's producing these 4 types of gametes. We've got this one over here doing the same thing, right? And now with sexual reproduction, we're going to put some of these with some of those, some of these with some of those. We're just going to mix and match randomly as to where those go. We're going to pile all the combinations up that we could, all right? Now, for many of us, the way we would have learned to solve that, and it's a very good teaching tool, is what's known as a Punnett Square, right? And you may have used your Punnett Square already for the monohybrids, but Punnett Squares start to get a little more complicated when we go to dihybrids or trihybrids, but they still work quite well. So Punnett Square would just be where you take the gametes from one parent, maybe this smooth green one here, okay? Traditionally the female and then the male over here, but doesn't really make any difference with plants particularly. You just take the gamete types that we saw over here. We saw some big S, big G, some big S, little g, all that. Write them down, right? However many there are. There's 2, there's 4, there's 6, you write them across. We take the other individual and write all of its gametes going down. In this case, we've got 4 types as well. And then we sit there and we work through the problem. We say, okay, we drop down big S from here, big S from there, big G from here, big G from there, and we put them down. Then we go, okay, over here now we have a little s coming down from this parent. We have a big S from this parent. We just write them in there. And we have a big G coming from this parent, and we have a little g from there. So we get a genotype of homozygous for the S allele, big S, but heterozygous for the G allele, big G, little g. And we keep doing the same thing. We just do it 16 times. We just keep dragging that through there. And when we do this, eventually what we're going to see, if we work at it, is that if we count all this up, we will end up with a ratio that looks something like this, a 9:3:3:1. How do we get that? Well, we start picking through all of these parts and figuring out how many of each type we have. We have one of these right here, so that one right there is actually where we get our yellow wrinkled right here, right? Because it's complete homozygote. For the S allele, it's going to make it wrinkled, and for the G allele, that's going to be yellow, all right? And that's exactly the Mendelian trait. We haven't have anything weird happen. And we started out with these sort of smooth green and wrinkled yellow original parents. One of those traits for each one, and one of the colors disappeared. The wrinkle disappeared, and the smooth and the yellow disappeared in the F1, and then they start to reappear back in the F2, just like they did before. Smooth is the dominant, so smooth retained itself in the F1. Wrinkled is recessive, it disappeared in the F1, but here it shows back up in the F2. Same thing with green. Green was dominant, so it shows up in the F1. Yellow disappears, and then suddenly yellow shows back in the F2. And you can keep doing the same thing, right? We start looking at, say, here. You say, okay, it's got big S, big S. It's going to be a smooth green. We say, oh, well, that's also going to be a smooth green, right? Even though it's got a recessive allele here, the G is going to take over, so it's going to be smooth for that, smooth for that. In fact, we keep doing that, we see that they're really all we need is one big S and one big G to be smooth green. And if we count up all those that we have, there's one, looks like there's another one, there's one, there's one, there's one of those. That each one of these has at least one big S and one big G. Doesn't have to have, 2 of them, but it can have that. And then finally, there's this one as well. And if there's another one, okay? So you keep going through here and you basically go, okay, I've got 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 of them give me a smooth allele and a green allele, the two dominants together. It looks something like that, okay? That's where I get this number right here. To get this 3 down here, I look for things that have a big S but 2 little g's. So there's one right there, okay? There's one right there, and there's one right there. And I just keep doing the same thing, working through it, okay? Now, this works fine. It's a great way to do it and learn how to actually do these Punnett squares. Very important. But it's not the simplest way to do it. Okay, it's a great learning tool, not necessarily the best tool. So what's an alternate tool? Well, let's think about what's going on here. What's going on here is our second principle, okay? And people get it's all confused all the time, but very simple. First principle is segregation. That's the one at the top here. That deals with one locus, okay? First one, right? Independent assortment is the second principle, and it deals with 2 or more loci. So you got a locus over here, you got a locus over here somewhere separate from it. Completely different chromosomes for our purposes, okay? So what does this say? It says during the formation of gametes, so once again we're back at meiosis, these segregating alleles, so this stuff that's going on here with segregation, they also separate from themselves independently. We're going to call that assorting. So they assort independently of each other. So what we're talking about here is we've got 2 traits, one wrinkled smooth over here controlling that, and we've got another trait over here that's controlling color, green or yellow, and whatever happens over here to separate these alleles is completely independent of what's going on over here, okay? Basically, they're separate loci, and for our purposes right now, they're on separate chromosomes, all right? Notice this word independent. This is happening completely independent of this. Let's think about a lecture that we hopefully just finished not too long ago when the word independent became very important, right? Why? Because you can produce these final assortment ratios, we're going to call them. In our case, this was a 9-3-3-1, but we'll see 3-1-3-1s and 1-1-1-1s and what else? We're going to see a 1-2 to 1-2 to 4 to 2 to 1 to 2 to 1 at some point, and all kinds of variations. Those assortment ratios, those things that involve 2 or more loci, can be generated by simply multiplying these segregation ratios together. Why? We apply the product rule. Very straightforward. So if you're trying to figure out genotypic ratios, just figure out what look on the Mendel's Big Six, figure out what they are, whatever cross you made over here, what genotype ratio you expect, and then multiply them out. If you're trying to do phenotypic ratios, then for right now, you're going to go over in this column. It's going to get more complicated later, but for right now, we're going to jump over to this column. All right, so let's go back and see if we can do this without a Punnett square. All right, so this is really the cross we're making, right? If we allow ourselves to just ignore the G's and worry about what's going on in the S locus, we're really crossing a het right there with the het right there. And very simply, genotypically, it gives us a 1-2 to 1. Phenotypically, it gives us a 3 to 1. Now we can ignore that and just look at the G locus, figure out which of the Mendel's Big Six that is, right? And it turns out to be the same. Okay. Now we figured out what's going to happen from both. By the product rule, we simply can multiply them together. What that means is we can take this one and put it together with that one and keep the numbers and multiply those numbers together to figure out what we're going to get, right? So genotypically, that's going to give us big S, little s, and all that, right? But big S blank for our purposes phenotypically, and big G blank, we're going to get some of those. And we're going to be able to take these numbers, okay, which are this, we're going to keep working at it, right? Got all 4 of the possibilities there. It could be more possibilities. We know what the phenotypes are because who's dominant, right? So big S is dominant, which is smooth. Big G is dominant, which is green. Big S, yellow. We got all those things right there. Now all we got to do is take the numbers and multiply them out, okay? So let's go back and see what I did. I said for the first one, I've got a 3 that goes with S blank, and a 3 that goes with G blank. So 3 times 3 is 9. For the next one, I've got a 3 right there that goes with S there, but I've got 1 right there. So that's going to be a 3 times 1. Next one is going to be a 1 times 3, which is also 3. And then the final one is that. We get our 9-3-3-1 ratio. Okay. So welcome to the Punnett squares. That's perfect. It's a great learning tool. But this will also work, and it'll make it very efficient. And if you understand this, you understand Mendel, you understand how to do it, and you're going to can work through it. Okay, so that's the first part. We'll move into the second lecture where we'll start looking at ways we can actually utilize this information and to actually take Mendel's principle of segregation and Mendel's principle of assortment and use those for our test.